Yes,
Yeah.
so hi everyone, this is Yiran, and it's my pleasure to cover the topic of CytomX. Uh, sh- um, Shared some promising phase one data for their masked ADCs in colorectal cancer. So the remarkable things about this news is, I believe this is the first masked ADC candidates to really show clinical positive or promising signals. CytomX is basically a platform company which has been the pioneer for developing all sorts of masked biologics, including masked bispecific. They have their own struggle on that front, but their prior attempt in masked ADCs, which use older generation of toxins and linkers, have been proved failed, which basically the program have been abandoned. Yet their Varseta-M, which is their newer generation of the Probody ADC, is designed to target EpCAM, which is a widely expressed tumor-associated antigen. The core innovation is use the mask to overcome the historical challenge of targeting a target like EpCAM, which is, of course, very dominantly expressed on the cancer, but notoriously widely expressed in the gastric tract. Which has been a very key factor limiting the development of the EpCAM targeting reagents. So with this masking technology, it has been shown preclinically to increase the maximum tolerated dose to sixfold in monkeys. And also with their newer generation of linker, is supposedly to increase the release of the mask in the tumor microenvironment, which has been known to be an issue in the prior generation. And also it adopted one of the most successful toxins among this ADC era, the topoisomerase inhibitors with a DAR of 8. Move to the next slide. When we talk about the target, as we know for target therapies, a lot of targets have the issue of the incomplete coverage or only apply to so few patients. Yet for a target like EpCAM, you are shooting for hopefully almost all comers, almost all patients with colorectal cancers and various other cancers that are carcinomas in the gastric tract are very highly expressed. You can see how dominant this signal is. Yet, it has been known with this target, the issue is on target of tumor toxicity. So that's why they hope with the masking, although it can also be recognized by EpCAM expressed on the healthy tissue, but with the relative high expression in the tumor and with the linker that only get digested in the tumor microenvironment, in that case, the binding to the EpCAM is only released in the tumor microenvironment, therefore give it selectivity. So that is the hope. And once it's being released in the tumor environment, being bound to the target, it increases half-life, therefore enriching the tumor microenvironment and increase the selectivity of the tumor delivery of the ADC. So let's move to the next slide to see how effective the strategy may work. So from the dose escalation studies, we see they go up to 10 mg. 10 mg is high for ADC. It's not like the six-fold to 10-fold high as they see in the preclinical model. I would say it's only moderately just one-fold at most higher than conventionally what other ADCs have been shooting for. So clearly, the masking strategy haven't been so remarkable in increasing the therapeutic window. And of course, we see a dose relevant associated response in the so their lowest, not the lowest, the 7.2 mg group, the response rate is already in the single digit, too low to be useful. And 10 mg is performing better than the 8.6 mg, and in this OR is as high as 30%, which is quite remarkable considering this line of treatment. But still, we have to stay cautiously optimistic, as we could tell. I mean, it's already struggling on this end, and in a greater population, can it replicate? We would stay cautiously optimistic. And move to the next slide. The duration of response is something noticeable in third-line refractory colorectal cancers, because normally for other, let's say, those kinase inhibitors, even patients respond, the PFS can be as low as 2 to 3 months. 6 months PFS is not bad in this line. But overall, my comments for this efficacy is on the borderline of promising to be worthwhile to move to the next stage of clinical development. Yet it also sits on the line that the OR can drop quite significantly, just considering the bias from so few populations. And we have to also keep in mind there are other many pipeline products being developed in this line, including ADCs to other targets, including, let's say, PD-1 IL-2s, other immune modulators, also showing OR approximately in the similar range. So the competition from other fronts is really high. The key issue for this drug is, can it proven to be effective? Can the therapeutic window prove to be acceptable? Next slide, please. So when we examine the toxicity profile, especially the diarrhea, which is the on-target off-tumor toxicity, the occurrence is really high. Overall, in all patients, it's up to 85%, and grade 3 is over 24%. So it's prominently toxic. So you cannot push the dose any higher. So you can barely tolerate the dose that are acceptably effective. Yeah, the toxicity seems severely limiting the it to go any higher. So that's the issue for those masked ADCs in general. Although the masking technology sounds promising for significantly increases tumor enrichment, while in reality, the cleavage of the masking is very slow. In the meantime, being an antibody, the unspecific uptake by the Fc gamma receptor is also quite prominent. So overall, I don't think they go the dose any much significantly higher than the naked toxins, and I think it's primarily limited by the unspecific uptake of the antibodies, therefore release of the toxins in the systems. So clearly there's so much need to be done to increase the tumor specific delivery with the current technology platform. I think is only marginally acceptable. Yet it is a promising signal, indicate we are heading towards a hopeful direction. So that is my overall comment for this topic.